Today, I'm honored to be here to present my current work on dry skin and a new strategy to treat dry skin. The dry skin is a pretty common condition characterized by the lack of moisture in the skin, affecting 30% of general population and as high as 85% of elderly population. The people bearing the dry skin will face the physical discomfort, such as tightness, roughness, flaking, even a vicious cycle from itching, scratching, and high risk of infection. The people will also suffer the psychosocial impact, such as embarrassment and anxiety. Even though the dry skin is not a life-threatening disease, it indeed impairs the quality of life. In the normal condition, these tiny glands, these tiny lobular glands, named as sebaceous glands, are responsible for producing the oil. And those produced oil will cover the outermost layer of skin to prevent transepidermal water loss. But under some abnormal circumstances, such as genetic defect or aging, we know that we are all aging. Sebaceous gland can be lost, and the oil production will be dramatically decreased, so there will be excessive transepidermal water loss, and eventually leading to the dry skin. So when we have the dry skin, we usually apply the cream. But we clearly know that the cream only treats the symptoms, but not the root cause. So what's the root cause of dry skin? Here is the answer: the loss of sebaceous gland. In my study, based on the evidence from genetic mouse models, we found sebaceous gland resident stem cells is a group of COX-20 positive cells. And those COX-20 positive cells is crucial for maintaining the integrity of sebaceous gland, since the deletion of COX-20 genes will lead to the loss of sebaceous gland, whereas the overexpression of COX-20 genes will lead to the multiple lobe of sebaceous gland. Mechanistically, COX-20, working as a transcription factor, can directly regulate the NOTCH1 gene's transcription. Here is a key question: can we restore the skin health? Actually, there is no chemical to regulate the COX-20, but luckily, there is NOTCH1 pathway activator, valproic acid. It may pave the way to resolve the root cause of dry skin and to treat the dry skin. In order to test our hypothesis, we create dry skin mouse models, and then we topically applied valproic acid onto the dry skin. 1 week treatment can successfully prevent, even revert, the loss of sebaceous gland and the onset of dry skin. And those valproic acid treated skin is comparable to healthy skin. This study displays a typical flow from bench to bedside. Firstly, we identify a new cell type with COX-20 positive cells, and then we explain the mechanism, the endogenous mechanism, and then we explore the preclinical application. Our lab is still on the way to restore the skin health. My name is Yumeng Zhang. Thank you for listening, and I hope my audience have shining skin.